Now let us discuss about the relationship between the microbial divisions. Though certain morphological structures, nutritional type, and other properties can be used for establishing the relationship between the microorganisms, but the more appropriate and more correct method of developing the relationship is nothing but using this 16 years fragment of ribosomal RNA homology studies, or sometimes the genetic composition. By knowing the complete genetic relatedness between the two organisms, we can firmly establish the relationship between the various species of different microbial divisions. Now here, this relationship between the microbial divisions can be studied by using this phylogenetic tree, or you can call this as evolutionary tree. Now here you can see this phylogenetic tree or evolutionary tree, which is constructed using something like 16 years fragment ribosomal RNA homology studies. What is the extent of similarity between one species to the other species when taken this into consideration of the 16 years RNA? Now what is the similarity and dissimilarity between one species and the other species can be known. The percentage or the extent of similarity and dissimilarities can be known, and that is how these phylogenetic trees are constructed. Now here you can see the phylogenetic tree is nothing but a branching diagram. It is in the form of branches, just like a tree contains the various branches starting from the same stem or stem root. Now this diagram also contains the various branches. It is a branching diagram showing this evolutionary relationship. So based on the evolutionary relationship, now a minor modifications or changes may occur within the 16s fragment of rRNA, and that is how there may be evoluting from the same common ancestral organisms. Now here you can see the phylogenetic tree mainly depends upon the similarities and differences in their physical and genetic characters, and in these genetic characters are very important. Now the taxa, mainly they are joined together in a tree which have the common ancestors. Now you can see here in this diagram. Now this is a simple diagram. These are the branches. You can see a simple branch and a common point which is from which the branches have been emerged. Now this common point is called as a node, and based on some differences in their 16 years homology, branching starts from here. Maybe this is one species and this is the other species. Now this whole now this node is called as a one taxonomical unit, and this complete taxonomical unit in fact has emerged from another common ancestral point, which is again a node. Now from here, now this branching occurs towards this side, and further branching may occur towards this side. Now this branching is based on these homology studies. Now that is how you are going to construct the phylogenetic tree, and based on the construction or based on the pattern of this phylogenetic tree, now we can know what are the relationship between the various microbial worlds. Now you can see here this phylogenetic trees may be different types of phylogenetic tree can be constructed. Now basically we have two types here: rooted trees. Now this is example of a rooted tree, now which is showing a common ancestral point at each node. Now that is nothing but a rooted tree. And unrooted trees where common ancestors are sometimes unknown. Maybe as far as 16 years homology studies are considered, there some species are related together, but the common ancestral point may not be known, and it is not usually shown in these unrooted phylogenetic trees.